I'm a postdoc at Dalhousie in Finley's lab, as well as working with Zvonimir Polyak at University of Guelph. And I'm located in Burnaby, BC, so suffering with the rest of the Pacific Standard Time people. And I have lived here for quite a while. I did my PhD, master's, and bachelor at UBC. So recently finished my PhD in bioinformatics there, where I studied in the Joy Lab, studying HIV. So let's get into it. Today we'll be discussing phylodynamics. The learning objectives today are to understand how and why we use viral phylogenetic inference. I'm going to be focusing primarily on viruses today. We'll define phylodynamics and phylogeography. It's important that we understand the basics of Bayesian statistics, and within that, comparing the coalescent and birth death processes. We'll discuss, and I want you to understand how molecular clocks are used to infer divergent states and convert trees to time trees. Then we're going to get more into some of the phylogeographic approaches using Bayesian and maximum likelihood ancestral state reconstruction. And then in the lab, we're going to put this together to apply both maximum likelihood in the Augur pipeline and Bayesian in the Delphi tool phylodynamic analyses to infer the time and place of origin of SARS-CoV-2 B117 alpha variant. And I think you will be familiar with some of this data from Emma's lab on the first day. Okay, let me just get rid of some of this Zoom stuff blocking my view. So to start off, I just wanted to kind of segue into viruses because I know a lot of the talks and labs have been around bacteria, but RNA viruses are unique in that they are measurably evolving populations. So evolution is happening on the same or similar timescale as transmission. So because they have small genomes, large populations, and they replicate very frequently and have these polymerases that are error prone, sometimes with a lack of proofreading, this leads to rapid evolution, as you can see in these plots. On the left, the RNA viruses are on the top left for their evolutionary rate. And over on the right, you can also see the time scale at which genomic variation is observed can be days or weeks. And because they evolve so rapidly, we can use them to infer viral phylogenetic trees that are useful in genomic epidemiology. Some of the ways in which this can be applicable is to estimate lineages or variants or whatever delineations, divergence times, and origins. This can be, within outbreak detection or to maybe put forth a travel restriction on a place where a variant has just emerged. It's also useful to designate nomenclature such as pango lineages and to track their frequency, to inform outbreak detection and support or refute epidemiological linkage. Also to compare selective pressure across genomic sites and lineages while considering that the tips you sample are not independently sampled. Further, to quantify groups' relative transmission rates or contribution to transmission and the rate at which they disperse. And finally, any of these rates that you're pulling out of the phylogeny, you could use to evaluate the impact of extrinsic forces. For example, non-pharmaceutical interventions like travel restrictions on COVID-19 importations or social gathering restrictions on the effective reproduction number. So this is sort of depicted in this cartoon here where we have a partially sampled transmission chain, which ends up being a partially sampled phylogeny, and from that we can deduce a change in the reproduction number through time. So let's just come to a baseline here of how we're using genomes and the shared mutations within them to deduce phylogenetic relationships. So say you have this small group of sampled hosts, well, viruses from sampled hosts, and the mutations that they share are colored. So we could see immediately that all of them have this green mutation. So maybe that's a mutation that occurred at a shared ancestor near the root. So we'll place that there. Next, we see that D has no other mutation. So it's sort of an outlier or outgroup. And then we have shared mutations for ABC and EF. We'll place those together. Finally, E has no further mutations, F has a singleton mutation, so we end up with that, and so on and so forth to generate a phylogeny from these shared mutations. Okay, but one must consider, what does this viral phylogeny, which was partially sampled from an epidemic, actually represent? And I apologize that these slides are familiar from Finley's presentation yesterday, but it is an important point. So transmission trees, pardon me, underlie phylogenies. So this is sort of a depiction of a transmission tree through time, left to right. And sampling is incomplete. This would be actually a relatively optimistic sampling. A lot of the time, sampling can be under 10%. And then we have the between host phylogeny inferred from these partially sampled tips, and it's really a best guess at the transmission tree. And it's even worse of a best guess because this is exacerbated by the enormity of tree space. So this is a table showing how many trees are possible for n number of tips. So this is the n are not large in this table. So we go from 4 to 136 tips. Once we get to 136 tips, 2 times 10 to the 267 trees are possible. So today, I think the tree we'll be working with is around 170 or 80. So you can sort of respect that we're not going to be, comprehensively looking at all the tree space. So our best guess. And this is also important to interpret in light of the fact that we are sampling viruses from an epidemic. So we're looking at a between host phylogeny. However, this also includes variation at multiple levels. So within cells, between cells, within the host, across different areas, and in some cases between species. So all of that variation and diversity is kind of being compressed into this between host phylogeny. And some of that variation, though, is limited. It's not infinite variation because of the transmission bottleneck. This is an excellent paper by Katrina Lithgow showing and modeling the size of that transmission bottleneck, where you might initially have a largely homogeneous viral population after being infected. Diversity arises within you, but only one or a few virion are typically transmitted. And those virions which are transmitted can vary from infectee to infectee. So it can be a bit of a complicated picture. So let's transition to talking about what is shaping the phylogeny. What are these underlying processes that dictate that? So there are a lot of them. The phylogeny is sort of impacted by evolutionary, epidemiological, and immunological forces. And I've colored these according to what I believed they were the closest to in category, but a lot of these do sort of span multiple. So some examples of processes that are impacting the shape of the phylogeny. So that's the shape in terms of the balance and the distribution of branch lengths. So we have within host evolution occurring, within that immune pressure. It's important for the virus to maintain infectivity and its cell tropism. The mutation rate, which itself evolves, is impacting the phylogeny. The absence of replication proofreading in some viruses, as well as the development of virulence, can be also selected for or selected against. And then we have some of these epidemiological forces, such as the generation time. So that's the time between consecutive infections. The transmission route. Is this an aerosol? Is this a sexually transmitted virus? Were there non-pharmaceutical or pharmaceutical interventions in place throughout its evolution or at certain times? Also, in the case of human RNA viruses like SARS-CoV-2, host migration plays an enormous role in dispersing the virus, as we'll discuss more. Then we also have social behavior to consider. Are people going to have a lot of contacts? Are they going to modify their social behavior upon learning that they're infected? We have population structure to consider as well, which is sort of the delineation of the population into more closely interrelated subpopulations. Um, and then we have some of these immunological forces, like whether people have been vaccinated and with what and how frequently, what their host immune response has been and how strong it is, and how much memory they have, and whether that wanes quickly. So these are the type of things that are considered in a phylodynamic lens. So phylogenetics are central to phylodynamics. I'll just distinguish these here. So phylogenetics is focused on reconstructing the ancestral relationships among a set of taxa, in this case viruses. And phylodynamics studies how all of these processes that we just discussed act and interact to shape a population's phylogenetic history. And this is the sort of famous definition from Brian Grenfell in this 2004 paper, from which these figures were also pulled. So let's spend a little time just making sure that we're all clear on what these figures mean. So on the left, this is a very famous figure actually, despite just being sort of a cartoon. This shows the immune pressure on the x-axis and the net adaptation rate on the y. So the black curve at the apex there shows that the maximum rate of adaptation occurs at intermediate levels of immune pressure, and that corresponds with intermediate strength of selection, not too much, where there's hardly any virus there, it can maintain a large population size, right? So the viral abundance as well is intermediate at the highest level of viral adaptation. And the way in which these different processes impact the shape of the phylogeny is nicely contrasted by these trees shown here. So these are trees for human influenza A virus, HIV between host population, and HIV within host phylogeny. So you can appreciate that these trees have different structures. One on the left, the human influenza A virus, you might describe as a bit more ladder-like, as well as also the HIV within host, where new variants emerge and they sweep the other ones from existence. So you don't have the co-circulation of diversity as you do with HIV between hosts, where you can see a lot of these tips are relatively close to the present. And also the distribution of branch length is kind of balanced across the tree, because these viruses have a very different evolutionary strategy with long chronic infections where they could, have a long generation time, as well as they're more structured geographically. So in North America, we primarily have subtype B HIV, not exclusively, but primarily. Whereas in sub-Saharan Africa, you're going to have a different mix of subtypes, mostly C and other recombinant forms. So just a quick look there. We're going to discuss more about how those processes differ in their impacts. So this is also shown here, where different processes, but also different population dynamics generate different tree shapes. So this is comparing when the underlying population size on the left has either an exponential or a constant, exponential growth or constant size, and how that impacts the shape of the phylogeny, where you have selection and balance to consider and structure across different population demes. So all of these things are impacting the phylogeny. And I maybe should have mentioned before, but a lot of the slides I'm pulling from inspiration from Taming the Beast, which Finley also mentioned yesterday. A lot of this material is available freely online, and I was lucky to attend this workshop in 2019 in Squamish. So that was a great experience. And among these processes, spatial diffusion leaves an imprint on the virus's genetic history and phylogeny. So now we've come to our definition of phylogeography, which according to this 2000 definition, more broadly, it is the study of the principles and processes governing geographic distributions of genealogical lineages. A more modern, viral-focused definition might be the study of the spatio-temporal distribution of viruses across the phylogeny. And here, some of the focuses of phylogeography historically have been whether genealogies or phylogenies are driven by the dispersal of viruses across hosts and geographies, or covariance, where their host and the virus are co-diverging together. And again, we can look at SARS-CoV-2 or influenza A virus versus HIV in terms of these dynamics, where SARS-CoV-2 has these broad rapid sweeps, as we saw when a Omicron emerged, for instance, basically wiped out all other pre-existing diversity. And then we have this disassortativity where it's mixing and dispersing across geographies. And it has that dispersal dominant pattern versus HIV, which has the coexistence of geographically structured subtypes, ongoing cluster transmission, and persistent chronic infections. We're going to see more of a covariance distribution pattern. Okay. So now that we've defined phylodynamics and phylogeography, we need to take a little bit of a step back and talk about Bayesian statistic. So I've just shown a couple of the tools that use Bayesian statistics for phylogenetics and phylogeography. You might be familiar with BEAST. So there's BEAST original, BEAST 1, and then there was BEAST 2 because some of the researchers involved wanted to branch off, pun intended, and develop their own packages. And now there's also BEAST X, which is kind of the replacement for BEAST 1. And there's also this new tool that we're going to be using in the lab today called Delphi. And it is a Bayesian graphical user interface that has it has made these analyses a lot more rapid through some tricky stuff that we're going to talk about later. Okay, so Bayesian statistics 101, you have Bayes' rule, right? And this emerges very simply from the rules of conditional probability. So the probability of A and B is equal to the probability of A given B times the probability of B, and so on and so forth with its converse. And when you equate those red and greens together and rearrange that formula a little bit, you derive Bayes' rule or Bayes' theorem, which is probability of B given A equals probability of A given B times probability of B over the probability of A. And we're going to break that down into what that actually means in terms of the phylogeny. So Bayes' theorem in phylogenetics, you can replace those with the model and the data. So Bayesian statistics is very much focused on the probability of the model given the data. And this is the posterior probability of the model parameters in light of the data. And let's also break down what these other things are in terms of their designations. So the probability of the data given the model, that is the likelihood. So a model which seeks the maximum likelihood is maximizing the probability of the data given the model. But in Bayesian statistics, that's just one part of the puzzle. Another very important part of this formula is the prior. So this is the probability of the model and all of the components that make up that model. And these are sort of like our beliefs or hypotheses about the functional forms that these priors should take. And those should be rooted in evidence because actually there's a lot of opportunity here to bias your model with improperly selected priors. And then in the denominator, we have the probability of the data. This is also called the marginal likelihood, and it's very hard to calculate. And a nice thing about Bayesian model selection is, so I'm just going to skip ahead one and come back, is that when we compare two models, one over the other, the marginal likelihood actually cancels out. So it removes the need for us to calculate that marginal likelihood. So that's a very nice feature of Bayesian statistics, and it allows sort of the underlying algorithm walking around this landscape to sample from the posterior to compare models and whether to select them or not. Okay, so the model itself is comprised of several different features, if you will. So the model consists of the tree, the demography or demographic population process, the site model or evolutionary model, the molecular clock model, and anything else that you want to impose on that, whether that's um, geography as well, would be part of your model. So let's discuss more how those are selected and what those mean. So we discussed this a little bit already. So the phylogenetic likelihood, this is an important point. And so the likelihood of the tree is based on the probability of observing the data under this specific model of evolution. This is a classic definition from Felsenstein, one of the founders of phylogenetics and likelihood methods. But in terms of the priors, so the probability of the model, these are again distributions of parameters chosen based on your beliefs and prior evidence. Usually a parametric distribution is going to be specified, whether that's uniform or log normal. And then sometimes this is just making a selection about a type of model. So for substitution models, how are the different nucleotides transitioning from one to the other? We have several choices, HKY, GTR, JC. These are all acronyms for defining the way in which those nucleotides change from one to the other. We don't really have time to get into that too much today. And then also priors can have priors themselves and to infinity. So hyperpriors are very much a thing. And parameter bounds are a part of that prior as well. Okay, so the demographic model. So this is part of our prior beliefs. So what is the underlying population demographics or species diversity, and how does this change through time? So this is also called the tree prior. And we'll discuss this a little bit more because it's actually a very important component in terms of what you think that underlying population demographic looks like. And typically you can sort of define these as being based on a coalescent or a birth-death process. And these are simply contrasted here, where you can see the coalescent is backwards in time, going from the most recently sampled tips towards the root as they coalesce towards the root. And then the birth death, sort of you start with your initial infected root, and births and deaths occur forward in time. Sorry, there is a lot of text on this tree, but there's a lot to understand about the coalescent, and I hope that you can take away some of these points. So the coalescent branching model is all based on the Kingman coalescent. It's describes the rate at which lineages coalesce, informing the rate of population growth. Because if you know how rapidly these lineages are coalescing, you can say something about the underlying size of the population at each generation. And the coalescent draws from sort of population biology dynamics like described by the Wright-Fisher model. You're tracing the ancestry of n samples back to the root. And then we also, it's an important point that we're calculating the probability of observing a given tree for a particular n or population size. But sometimes you'll see this written as NE. NE is the effective population size, because the true population size really isn't known. It's only that which is underlying the phylogeny, which we're going to be able to infer. And there are some important assumptions here that you might find problematic. I do sometimes. The coalescent assumes that mutations are neutral, which, some mutations are not neutral. They have positive or negative selection, but that is an assumption. It assumes no recombination or sampling bias. Again, debatable. and the underlying susceptible population is described by the specified model. So we can at least hopefully get that one right. So some of the different coalescent population demographics you could see here. So when the population size increases, like lightly exponentially versus decreases, this yields different underlying populations under that phylogeny there. So if you see more frequent branching and small branch lengths, that suggests a smaller underlying population size because those pairs of tips are able to coalesce more readily together versus if there's a large population size. And ultimately, through the coalescent, we infer the effective population size through time, as shown in this sort of landmark paper by Alexei Drummond, who was one of the founders of BEAST, showing how the population size of hepatitis C virus sequences went through time there. Okay, a couple more notes about the coalescent, and then we'll move on. So the other thing about the coalescent population models is that you can make them parametric, such as exponential or constant, and these were sort of the early derivations of this model. Or you can use a non-parametric model for the effective population size. So these are usually piecewise models, piecewise constant, piecewise linear, sometimes piecewise exponential. And then they usually also apply some smoothing, for example, using the Gaussian Markov random field, GMRF. So within that, some of the popular and easy to confuse models are the Skyline, Skyride, and Skygrid, which basically differ in how they're separating time into discrete bins. So in our lab today, we're going to be using the Skygrid model, which basically can separate them into uneven categories, which is nice because sometimes you want a category to be a bit larger, and then it applies this GMRF smoothing. So the paper to look at if you're interested more would be Gill et al. 2013. All right. Okay, let's discuss birth test sampling models a little bit, and then we can sort of set these aside. So this was the other type of population model where we're looking at a branching process forward in time. So in a birth death sampling model, you can think of birth as being infection or speciation if you're looking at a macroevolutionary tree, and then the death rate. So in a pathogen context, this would be recovery or removal due to sampling or death. Or in a macroevolutionary context, this would be extinction. And then we also have in some birth death models, incomplete sampling or also the fossilization process. And yeah, the birth death model has a long history. The first derivations were from Kendall in the 40s. And then some of the more landmark papers to note here are the incorporation of the evolution of a binary character through time. This was work by Wayne Madison, who is a Canadian. And then some other important contributions were from Tanya Stadler. So the birth death sampling with incomplete sampling, as well as the often encountered birth death skyline model, which is reparameterized so that you're not sampling from birth, death, and sampling rates in the Bayesian process, but you're actually looking at the effective reproduction number, birth over death, and also the become uninfectious rate, which is the removal rate, basically. But there are issues with that model, and I don't quite have time to discuss them today, but I was involved in a collaboration with Stylianos Luka and Matt Pennell that showed that these models are actually not identifiable. So you cannot simultaneously infer birth, death, and sampling rate without already knowing one of those rates' true values. So something to be wary of if you are thinking about applying these models. And some of the assumptions to be aware of here are that, well, they all assume that lineages are interchangeable, mutations are neutral again, and that there's random sampling. But the exact specifications of this model vary in their assumptions in terms of sampling, as we talked about, whether there were mass extinction events or concentrated sampling efforts, as well as whether sampling results in removal and some tree conditioning around the root. So these are all nicely summarized by a review by Eileen McPherson, another Canadian, although dual citizen, based out of SFU. And this is a review that I contributed to with her. Okay, so let's get into talking about the molecular clock model. This is a key concept. So the molecular clock model is the rate at which evolution is occurring. And typically mutations are accrued in a slightly clock-like fashion, although that can be strictly clock-like or in a more relaxed manner. And specifying a molecular clock model allows us to convert a genetic distance tree or divergence tree into a time tree. And the sort of thing to keep in mind here is that the genetic distance is equal to the molecular clock rate times the amount of time that has passed. So let's have a quick look at how this might work. So this is an influenza tree from Trevor Bedford. And initially, trees are typically inferred using divergence from the root in terms of substitutions per site. You could also have this just substitutions or mutations if you multiplied by the width of your alignment. And if we look at the rate at which these sequences are evolving, this is typified by the root to tip regression. So the RTT plot, where on the x-axis we have time, on the y-axis, sorry, my throat's a little dry. We have substitutions per site, so that those were our units from the prior tree. And the slope of this root to tip regression, which is just a linear regression, gives us our estimate of a strict molecular clock rate. So in this case would be 4 times 10 to the negative 3. Should say substitutions per site per year is the typical unit of the molecular clock rate. And this, I would say, is a very nicely fitting line. This would probably have a high R squared value, very little deviation from this plot. In reality, a lot of root to tip regression is not this clean, but you can quantify that. And so using this molecular clock rate assumption, we can convert this divergence tree to a time-scaled tree, which does look a little bit different. Sometimes you can have compression or expansion of some of those branch lengths. So yeah, here I wanted to note that the clock rate may be strict or relaxed, where we might have a mean that is described by a log normal distribution. And you can also either fix the molecular clock rate or estimate it. And also there are different things you can do in terms of assuming there's one clock homogeneous across the entire tree, or you could have a local or autocorrelated clock that kind of evolves across different lineages. So there's quite a bit of flexibility there in Beast in particular to specify that. A little bit on the strict versus relaxed molecular clock. So we've talked about this a little bit already, but just in terms of sort of looking at what this would look like. So this would be a depiction of a relaxed molecular clock where we have a range of values that are drawn from that log normal distribution, and those are applied on our sampled time trees. Yeah. Okay, so in using molecular clocks to scale our trees to the unit of time, we can apply this to conduct divergence dating, which is basically estimating the time of the most recent common ancestor, TMRCA, in order to estimate what is the origin time of whatever group of taxa you're interested in. And a good practice is to not just report a central mean or median value for this, but to always include a confidence interval, whether that's a highest posterior density, HPD estimate in a Bayesian framework, or if you're using frequentist or maximum likelihood inference, this can be a confidence interval across bootstraps or across resampled branch lengths in the tree. So this is a figure from another Alexi Drummond paper from 2006 of influenza A viruses. And you can see the yellow bars show that HPD confidence interval on the origin of any group of those taxa. So we could look at, for instance, the date of that root, which spans sort of somewhere around 1975 to 1980. So in more modern contexts, this has been applied to look at, for example, the origins of SARS-CoV-2 lineage B117, the alpha variant. So this is an excellent paper by Verity Hill and group and the Rambo lab, where they looked at B117 as well as background lineages of its parental B1 origin to look at, first of all, whether there was a different molecular clock rate or a different slope between those two groups, which they found, oh, even though they form two different, clusters, if you will, of data, the slope is the same. It's just that there was something that happened in between that was strange. So what is predicted to have happened there was that there was a chronically infected patient who had SARS-CoV-2 for a long time with intermediate immune selection, possibly in the presence of treatment, which can really put a lot of evolutionary pressure on the virus to diversify without it having such a functional immune system as to tamper that down. So the main point is, though, that the molecular clock rates themselves are similar, despite them being separated by that likely chronically infected patient. So they used this to estimate the effect of population size through time, as well as the growth rate pre-lockdown, lockdown, and post-lockdown of those two different groups, where they found that the B117 lineage was not, or it had a higher growth rate than the background lineages at all of those different times. And then they also interpreted the sequences per day. They estimated the effective reproduction number using the birth death skyline model, which I have issues with because of the non-identifiability issues. And they looked at the sampling proportion. But they did also date this origin of the B117, and we will discuss that more in our lab. Okay, so let's move on to the substitution site model. With the site model, so this is a combination of our substitution model, which we alluded to as being, for example, the HKY or GTR model, which are basically ways of specifying the underlying instantaneous rate matrix. And then the site model upon that specifies how that substitution model or transition rates vary across different sites in your genome. And a lot of the time you'll say in your priors that you want to specify this, and you might discretize a gamma distribution with a long tail for this site variation, and you want to say, well, there's probably some proportion of invariant sites there. So substitutions are modeled using Markov processes. So specifically, we use stochastic models called continuous time Markov chains, or CTMC, in order to model transition between these discrete states. So these discrete states in this case are nucleotides. And this is described by the rate matrix Q. And we also assume every site is evolving independently and that substitutions at each site are governed by this typically reversible Markov process. And this is a very central point because not only can site models and substitution models be used for nucleotides, but also this is the fundamental premise of a lot of phylogeographic methods because site models can also be applied to morphological traits, the roots of words. You can use phylogenies for languages or beers, I've even seen, as well as geography. So you can think of the geography, if we go back to this slide quickly, as the T, C, A, and G being replaced with all of the discrete geographies that you're interested in. So you have a rate matrix transitioning between those discrete states. So in practice, we might want to infer ancestral states such as geography from our observed tips, where we don't actually know what the state is at these internal nodes, but we have some idea of the transition probabilities and rates. And we use this to infer for our ancestral state using either parsimony, maximum likelihood, or Bayesian statistics what the probable state was at those internal nodes. So this is fundamentally how phylogeography works. I wanted to include quickly the different Bayesian phylogeographic methods that are available. A commonly applied one, even though it's from 2009, is the coalescent with discrete trait diffusion. So it's basically a Brownian diffusion process, and we're treating discrete traits as stochastically evolving characters, as we just described. Sometimes you'll hear this referred to as the mu-gration model, and that also is the case in the maximum likelihood case. And then we also have more strictly Bayesian phylogeographic methods, such as the travel-aware coalescent with unsampled tips, the structured coalescent approximation, as well as the structured birth-death. So that's similar to what we were looking at before, but you're essentially specifying more compartments for the population structure between those groups. And in the Bayesian phylogeographic framework, you are simultaneously inferring the tree and all of those other model parameters. In practice, that makes it very slow. So that's the biggest downside to these like formally but Bayesian phylogeographic methods is that even for a couple hundred tips of say 30,000 base pairs, which would be the size of the SARS-CoV-2 genome, that could take you a couple weeks to run a few Markov chain Monte Carlo steps there. So some applications of the Bayesian phylogeographic methods in the literature. There's so many, but this is just one example of using it to reconstruct the origins of the HIV-1 epidemic in Western Africa by Faria. And another important approach you might consider is to use likelihood-based methods. So here we're inferring the ancestral states and model that maximize the probability of observing the data using models that account for, evolutionary rates and branch lengths. Some of the landmark papers in this approach for the inference of binary and multiple node states. These are sometimes called BISA or MISA models, not the best acronym. And again, we have some Canadians here. Wade Madison, Sally Otto, Sarah Otto out of UBC was a critical contributor here. And there's a few different methods you can use to apply likelihood-based phylogeography or character evolution models. One of them is tree time, and we'll be using this in our lab today through the Augur pipeline. And this is a way to infer ancestral characters as well as ancestral sequences on time-scaled phylogeny. Another method that I have used in my own analyses is through the R package ape. The ACE command, which is short for ancestral character estimation, is also a nice way to do this relatively rapidly for a large number of categories, because that is another thing with the Bayesian phylogeographic approach is it is limited in the number of discrete categories that it is able to converge on for that rate matrix. Whereas in a likelihood approach, you can include quite a few more, up to dozens of different geographies in your phylogeographic inference. So here's a nice example of using tree time through the Augur pipeline for phylogeographic reconstruction. So this is for Ebola virus in Democratic Republic of Congo, DRC, in 2018, from Kingata Lusamaki et al. In Nature Medicine. And you can see how we have all of these different regions in DRC as colored in the legend, which we can apply to infer the ancestral origin, which I believe they found to be Mombasa. So as I mentioned before, you can use that continuous time Markov chain discrete rate transition matrix for any evolving trait. So this could be, again, geography, mutations, host species. So say host species, if you have a virus like H5N1 or H3 influenza A virus that is rapidly and frequently switching species, you could use this approach to reconstruct the host transitions. We could also look at drug resistance phenotypes or hospital outbreaks. So anything that is evolving across that phylogeny and leaving an imprint on the phylogeny is up for grabs here. So on the right would be an example of using this for a hospital outbreak, where you might see, oh, well, we have 3 sequences from a hospital. Are these related to the same spillover from the community? This phylogeny here would deduce that no, actually, those were 2 separate spillovers into hospitals. And here is an example of using this to estimate the occurrence and timing of host jumping events. So this is from Brad Pickering et al., who works out of Winnipeg for PHAC or FAC, and Finley was a co-author on this as well, where they were looking at how SARS-CoV-2 variants had basically transmitted from humans into minks and then into deer, humans, and how that happened and exactly what the timing was for that. So I direct you towards this paper if you would like to learn more. Okay, another application of phylogeography that I have used in my own analysis is to quantify viral importation rates through time from different geographies of origin, and also to identify domestic sublineages which were imported from abroad. So this is depicted here, where phylogeographic inference of viral introductions includes both sublineages and singletons. So sublineages in this context for SARS-CoV-2 are international viral introductions resulting in sampled transmission. So they might also have, spilled back into another country. And then we also have singletons, which are international introductions with no further sampled transmission. And I used this approach in my PhD research, which we published this in eLife in 2022, to look at what were the sublineages of the first 2 waves of SARS-CoV-2 in Canada. We found that there were a lot of introductions in the first 2 waves, and this was in the midst of non-pharmaceutical interventions. However, despite there being over 2,000 introductions, less than 1% of those introductions led to 77% of the sequences. So it was like an over dispersed epidemic, as we say, where there's infrequent super spreading events. And the origins of these SARS-CoV-2 sublineages were diverse. The USA was the largest introduction source in the first and second waves, but there were contributions from a lot of countries. So the diagram on the left is showing, it's an alluvial diagram showing the flow from global origin to Canadian province, colored by Pango lineage. And then I also looked at the size of these sublineages in the first and second waves. So we actually saw that there was a turnover, turnover from the first to the second wave, from one set of sublineages to a different set of predominant sublineages with the first detection of the alpha variant B117 in the second wave of SARS-CoV-2. And I looked at this introduction rates through time sorry, this came out weird with the PDF, but how did this importation rate change in the midst of high and low stringency of non-pharmaceutical interventions such as travel restrictions? And we found that in March 2018, when we entered this period of high stringency with the borders essentially shutting down, this resulted in a tenfold decrease in the introduction rate within 4 weeks. Although it did not eliminate introductions, we still had that low level seeding the second wave. Okay, so that was a quick segue. Another thing I wanted to talk about, I think I have 10 minutes left here, is how we can use phylogenetics in sort of a phylodynamic context to quantify and detect evolutionary selection. So one way you might do this is that Tajma's D captures deviation from neutral change. So you can compare the singleton mutations to their shared diversity. And this is shown here on this spectrum on the x-axis where we have 0 sort of deviation from neutral change versus positive and negative selection. And one way that you can detect selection and do this in sort of a phylogenetic framework is using the DN-DS ratio. So DN is your non-synonymous mutations normalized, and DS are your synonymous mutations. So if this is not a clear concept, so a synonymous mutation would be a nucleotide substitution that results in the same codon, so it doesn't actually impact the phenotype, versus a non-synonymous mutation is actually going to change your codon in your amino acid. So by looking at the ratio of non-synonymous to synonymous mutations, you can tell whether something is under neutral selection, if those are about the same, or if they're greater than 1, then we have more non-synonymous than synonymous. This is going to be a case of adaptive, positive, or diversified selection. Those are all the same thing. And then if the DNDS ratio is less than 1, so there's actually more synonymous than non-synonymous mutations, then we can sort of deduce that there's purifying or negative selection at play here. And some of the challenges with this are that, yeah, mutation rates are going to vary over time and across different areas in the genome, and that genomes are interrelated and those mutations are not independent. So that's why it's important to use the phylogeny in quantifying that. And as you may be highly aware, mutation rates do vary across the genome. So this is a look at SARS-CoV-2 genome with the y-axis showing the variation at these sites. So in the ORF1b gene, which is the blue region, there's actually very little variability, relatively speaking. So this would be under probably purifying selection because it's conserving functionality there, or there's some other probably a functional conservation at play there. Whereas in Spike S, we can see some of those loci are highly variable. And this is subject to probably, and I think evidence suggests diversifying selection. So because there's immune-driven selection for Spike to evade antibody detection, and there's other evolutionary pressures in terms of the junction site between its S1 and S2 subunits and other features like that. So um, yeah, we can look at the non-independence of events in related genomes in this way. So mutations are shared by closely related genomes. Therefore, genomes shouldn't be treated just as independent observations. There is definitely potential for bias there. If you're ignoring that, you can think of it as akin to ignoring temporal or spatial autocorrelation in a model. So for instance, you can see that A, B, and C all share that mutation, but they have a different number of synonymous and not synonymous mutations. And one way which we might formally use a hypothesis test to look at this, what regions and tips are under adaptive selection would be using the branch sites random effects likelihood. So is there a significant proportion of sites within selected branches where the DNDS is greater than 1 would be the type of question you could ask with that. And one more thing I wanted to talk about on this topic is you could also use this approach to test, for example, for remdesivir resistance selection. So remdesivir is an antiviral that is used to inhibit the replication of coronaviruses such as SARS-CoV-2. And you could use, for example, ancestral state reconstruction to look at where remdesivir resistance might have occurred in the ancestral states, but you can also use it to group, you can group these by their relatedness in terms of the underlying mutations there. So, okay, I think I'm wrapping it up here. So I did want to discuss what the limitations are for phylogenetic analyses and how this impacts how we can interpret it appropriately. So there are always a lot of biases when we're dealing with empirically sampled data, especially from public repositories, in terms of non-random sampling. So in a perfect world, everything would be surveillance sampled at random, but in practice, you have a higher tendency to have sequences from outbreaks, hospitalized cases that are severe, and for that reason, also usually older age groups who are more likely to be connected with care and have bad outcomes with comorbidities, as well as changes in testing criteria can very much change the reason for sampling underlying your sequences. So keep that in mind. There's a lot of sources of uncertainty in terms of the sequence inclusion, whether that's through non-random sampling or through subsampling. There's also sequencing error to consider, a lot of uncertainty from the tree inference process because we cannot comprehensively look at that entire tree space. We also sometimes have identical sequences that result in polytomies where instead of having a bifurcating tree, you have a giant branch with a bunch of sequences connected with no mutations, and the model assumptions that we impose. For instance, both the coalescent and birth death model assume that mutations are neutral. So keep that in mind. It's challenging to appropriately structure your subpopulations. I know this is sort of vague, but it is an important point. So if you're talking about discrete phylogeography, how are you grouping those sequences into those categories and what bias might that impose? Something to keep in mind. And there's a lot of opportunity in Bayesian statistics in particular, but also in likelihood approaches to misspecify your model and your priors. So whether this is the evolutionary clock rate, the serial interval, which is similar to your generation time, reproduction number, dispersion, symmetrical migration rates, all of these things need to be considered in interpreting phylogenetic analyses. To summarize, pathogen, evolution, and epidemiology are intrinsically linked. Phylogenies are structured by multiple processes, both all of evolutionary, immunological, and epidemiological, and those leave an imprint on the tree. Phylodynamics applies either Bayesian or maximum likelihood phylogenetic models, We're not going to discuss parsimony because those are not widely applied. And we can use these approaches to reconstruct transmission dynamics, infer the timing and location of outbreaks' origins, and estimate epidemiological parameters such as dispersal and the reproduction number, and to test for episodic selection across the tree. Okay, and with that, I just wanted to thank the Canadian Bioinformatics Workshops and Pathogen Genomic Epidemiology team who put this all together, the instructors and the students, for all of you for being here. And thanks to Finley for some of the slides material that I borrowed, as well as the instructors from Taming the Beast, in particular, Alexi Drummond, Louis Duplessis, and Trevor Bedford. I used a couple slides of his as well. So yeah, thanks for listening.